Structure of cell. Let's think about the cell development. Maybe the cells started from water environment. It could be seawater. And to isolate the life space from environment, they utilized phospholipid. Cell invented the gene, and then gene is the most important material. That's why they protected the gene with a nuclear structure. Later, they invited the mitochondria for efficient metabolism. Also, lysosome was made to make the garbage processing other unit. Also, the cell made ER or Golgi-like structures. These are the general concept or the general structure we believe the cell look like. However, if you look at this kind of optical or the microscopy images, you can realize that it's very difficult to see the small structures inside the cell, such as the nucleus. This is mainly because the diffraction limit of light in optical microscope. Then how do you know whether the cells actually have those kind of a structure? Well, through electron microscope technique, at least now we can see the nuclear structure or mitochondria structure through the microscope's super high resolution power. So, optical microscope is quite difficult to see the small structure inside the cell. Well, maybe the only exception is maybe chromosome. Chromosome is a condensed DNA form, and then the word chromosome means chromo is a color, and then zome means body. So that means chromosome is a colorful body. So DNA or the chromosome is known to be easily stained by colorful materials. So even in 19th century, the scientists could observe the DNA structure through microscope. But this is not natural the images. This is the stained images by some colorful materials. So in this example of onion root cell has an actively dividing cell population. So that's why many cells are in the interphase state, but some cells actively condense the DNA and actually showing this kind of active cell divisions. So this is kind of a starting point of cellular imaging.